Hello class. This week we are going to cover a photosynthesis lab. You're going to go into a lot more detail on photosynthesis in the lecture part of class, but we're just going to do a quick overview of photosynthesis through a laboratory experiment. We'll go through an experiment that you can do at home, but first let's talk about photosynthesis itself, right? We know that living organisms, systems, etc., we require free energy and matter in order to grow and reproduce. In plants, this is the process of growth and development, right? Energy deficiencies are not only harmful to an individual organism, but they can cause all sorts of challenges at population or ecosystem levels. And therefore, organisms have evolved to develop strategies that have been conserved through evolution, right? We capture, we use and store free energy. This process in plants specifically is photosynthesis. Now, photosynthesis occurs through a set of enzyme-mediated steps, or these enzymatic activities that capture light, solar energy. They build energy. They build these carbohydrates. They develop these glucose molecules, right? That the plant can use. It's plant food at the end of the day, right? We see that through this equation here. Photosynthesis uses carbon dioxide, water, and light, and it produces our energy-rich molecule and gives off oxygen, right? That's how we interact with plants. And this is something that should sound pretty familiar in a 400 level college class, right? So at this point, I'm making the assumption that you get the gist of photosynthesis, at least for the purposes of this class. And in general, we can measure the rate of photosynthesis through either the consumption of carbon dioxide, this first part of the equation, or the release of oxygen, the tail end of the equation. In this lab, we're going to talk about what sort of factors can affect the rate of photosynthesis. We're going to talk about how we can determine the rate of photosynthesis in a simple at-home experiment. Now, if you've done this experiment before, I'm sorry. If you haven't, we're in for some fun. Now, let's first talk about some variables that can affect photosynthesis itself. We can think about environmental variables. This could be light intensity. This might be having a 20-watt LED compared to a 1,000-watt HPS bulb sitting over top of a plant, right? There's going to be a lot more light given off by that 1,000-watt HPS bulb than there will be a 20-watt LED. So the amount of overall light, that could be something like 2,500 moles of light, right? Compared to maybe 100. We can talk about light quality, the color, the wave bands that are actually hitting the leaves. If we're talking about having all green light hitting the leaf, that's going to be entirely reflected, right? This way, leaves are green. That's the light, the color of light they reflect, the green wave bands. But if we hit that leaf with more of those red-blue wave bands, that's going to be absorbed, right? So the light quality affects the rate of photosynthesis. Could be temperature. We can maybe think about how plants tend to grow in colder temperatures. I don't know. I'm from the Midwest where it gets pretty darn cold, but we tend to see this in winter up here in Chico too, right? Plants don't grow as much in winter, right? That's why we see a lot more growth and plant activity happening in summer. Could be carbon dioxide, right? 4,000 ppm or 400 ppm carbon dioxide versus a 4,000 ppm. It's going to have some huge effects on the rate of photosynthesis and plant growth. We can think about plant variables, right, that affect photosynthesis. We can think about the leaf chlorophyll concentration, the active home site of photosynthesis. You can think about leaf size and the plant's ability to capture light. That's a lot of plant architecture. We can talk about stomata density and distribution, the plant species itself, the age and relative activity, the enzymatic activity that takes place in each leaf. the type of metabolism, and so forth. Okay, so this lab is going to talk about or go over an investigation of photosynthesis. We talked a little bit about what it is. Now we're going to build a little bit off of what we know. In this case, we're talking about the use of oxygen, right? Instead of the consumption of CO2, we're going to focus on the production of oxygen, which allows a leaf disc to float. And look at this in a light and dark reaction system, right? So with that, I'm going to ask you to take a minute, track down these materials. You'll need your cell phone as a timer, spinach leaves. We've got some in the fridge for lunch tomorrow. A hole puncher. And it's pretty important to have it. Ripping leaves will be a little bit tricky. Hole puncher works well since we can use that smaller size with the syringe that you all have already ordered. 2 clear plastic cups are preferable. If you don't have those, a glass cup maybe. If you have a a wine glass sitting around, that's fine. A lamp, about 100 mils of water, baking soda. Now, baking soda is going to be our source of carbon dioxide, right? Carbon dioxide, that's the first input, and 2 syringes. So take a minute and track those down. Okay, so the experiment that we're going to do is based off a procedure that uses a vacuum, that's the syringe, and the syringe actions to create a vacuum. And this vacuum is going to remove trapped air in the leaf. It will suck the air out, right? That's the gases. In a solution that has, in this case, we're calling it baking soda, but it's bicarbonate ions. This is our carbon source for photosynthesis. Now, with these gases removed from, as an overview, removed from the leaves, those leaves will sink in that bicarbonate water solution. And our hypothesis is that when we place these leaves that have been evacuated of air in a sufficient light area underneath the lamp, we can speculate that the photosynthetic process will produce oxygen bubbles, and these oxygen bubbles will change the buoyancy, the buoyancy of the leaf disc. It'll cause the leaf disc to rise. And we can speculate that if we leave them in dark, that's more of our control, right? If we leave these leaf discs in the dark, we remove light from the equation, right? Let's hop back up to the equation here. If we remove light from the equation, those leaf discs won't rise because oxygen is not produced and that photosynthetic process does not occur. Okay, so at this point, I'm making the assumption that you found your materials. We're going to work through. So step 1, we're going to fill 2 cups. These are the type of cups that I'm using for this. Got some sitting around from previous use. We're going to fill these with about 100 mils of water each. And I'm going to put about a pinch. If you can be specific, weigh out a gram. Not everyone has a scale for baking their sourdough loaves, so just put a little pinch of baking soda in each of these cups and stir it pretty well, right? We're making the argument that, if you have a little bit more in one cup versus the other, it's not the end of the world because we're taking out a key factor of photosynthesis. We're taking light out of the equation. So just a good guess of about a pinch of baking soda in each cup is fine. Now, I want you to use a hole puncher and punch holes in your spinach leaves. You'll need about 20 leaf discs in total, and they should have avoid big leaf veins. So a leaf vein like this is fine, but if you have a really big midrib vein, that's going to be a little bit detrimental to this work and give you a harder time. So take a minute, punch out those 20 leaf discs. Maybe if you pause the video right now and put 10 in each, just 10 for each cup. So we won't put those in there just yet. Just punch out a total of 20, set them aside. Okay, so you've got 2 syringes, right? I'm going to ask you to put 10 leaf discs in one syringe and then 10 leaf discs in the other syringe. It's pretty important that we have 10 in each. Otherwise, our math won't really add up. Once you put those leaf discs in the syringe, add about 5 mils of that baking soda water from the beaker into the syringe. So take a minute to do that, and you should see that that back cap will pop off. Go ahead and pull that open. Put the spinach leaves in that back cap area, put your water, and you're set. Once you have that, lift your syringe upright like this and evacuate what's left of the air in there, right? So kind of like what you see with those weird doctors in the cartoons or the movies where they get a little bit of that water released by pushing that syringe up, that little end bit here, and getting that water pretty high to the top, right? Okay, now this is the technical part of this project. You want to put your thumb on the top of the syringe and then slowly pull the plunger down 6, 7, 8 times and shake it along the way. Make sure those leaves are kind of floating around nicely, those leaf discs. What we're doing is we're creating that vacuum. We're evacuating the air from the leaves. Now, the YouTube video I posted does a really good demonstration of this step. And I'm going to ask you to complete this step for both syringes so that you see at the end of it, your leaf discs should not be at the top. They should sink, right? Not floating. If your leaves have been evacuated of air, they should be at the bottom, sunk. All right, so take a minute and do that. Okay, so I'm making the assumption that you've done a pretty good job on that step. Let's keep going. Now, I want you to go ahead and make sure that your lamp is plugged in, turned on, and those photons are jumping around the place, right? Have one lamp on, then have a nice area identified for your dark conditions, something that is about room temperature, so don't put it in your refrigerator or freezer. Maybe put it in a cabinet, right? Somewhere where there's no light available and you can take a peek at it pretty easily. So go ahead and take a second to get that step completed. Okay. Now I want you to take another minute here and empty both of those syringes that have been that have the leaf discs that have been evacuated of air from that mesophyll layer, and empty them into their respective plastic cups, right? So at this point in time, you should have 10 leaf discs per cup still, 1 syringe per cup. Once you have those 10 leaf discs in each cup, it's time for the action, right? At this point in time, we're going to place 1 beaker underneath the lamp, that beaker that has those 10 leaf discs in them, and the other in a dark place. That's up to you to find. Now, once they've been placed there, I want you to take about 17 minutes to count and write down the number of leaf discs that rise to the top of the cup after each minute. So we'll look at this in the activity sheet and go over what this exactly looks like, but you should see that this is a cumulative recording, right? As opposed to the number of new leaves that have risen. You're taking a cumulative note. How many are at the top, or conversely, how many have not risen, right? Repeat this for 17 minutes. Do 1 count of floating leaves per minute. If you find that you have 11 leaf discs floating, you might want to double check things because each cup should only have 10. So you can complete this activity with your own data, or you can use mine. With that, I'm going to pull up the activity sheet. And here's what we've got. Here's the sheet that I'm using. Now, You'll see that we've got a column for minutes, and 17 minutes in total, and a number of floating leaf disks. Again, we start at 0. None of your leaf disks should be floating. And you'll see that eventually, in these light conditions underneath the lamp, after about 9 minutes, we got 1 leaf disk that floated, 3 in total after 10, 5 after 11, 8 after 12, and finally all of my leaf disks were floating after 13 minutes, and we don't go over, we just stick at that 10 part. A little bit of a different story for those dark conditions where we've only got these 1 leaf disk that surprisingly rose after 15 minutes. So go ahead and take those recordings and then complete this activity. Now it's important to note that when we're doing this, we are indirectly measuring the rate of photosynthesis by the rise of that leaf disc. So that leaf disc that we chopped out, that still has ongoing photosynthetic activities happening, right? That's where that oxygen source comes from that causes the leaf to rise. There's no other way for that leaf disc to rise. So make sure you have all of that air evacuated. And there's a lot going on in this whole reaction, but you need to just remember that cellular respiration is taking place at the same time as photosynthesis. We have these two processes occurring. Aerobic respiration, this will consume oxygen that has accumulated in that spongy mesophyll layer. So these two processes will counter each other with respect to that oxygen accumulation in the leaf discs and those air spaces in that spongy mesophyll layer. So this is a relative estimate of photosynthetic rate through this buoyancy of the leaf discs, right? Fun experiment. Go ahead and in this first question, put that line graph in. It should just be one graph. Make sure your axes are labeled. Make sure we know what color each line represents. So if you have a line for light, I don't know, make that red. If you have a line for dark, make that blue. Whatever you want to do on that end is fine. I want you to tell me what this graph tells us about the necessity of light and that photosynthetic reaction. And I want you to think about this a little bit differently. Instead of looking at light, what other variables could you measure that are crucial for photosynthetic rate? We talked about that a little bit in this video. So if you aren't sure what those other variables might be, recommending going back a few minutes towards the start of this video. And then tell me how you would conduct the experiment to test that hypothesis. That's it for this week. Thank you all for taking part, and I will see you pretty darn soon. Thanks.